This is a picture of a human cell. As you can see, the boundary or the border of the human cell is the cell membrane. Inside the cell membrane, you have there the space which we refer to as the cytoplasm. The largest organelle of the cell, the one that is most of the time situated in the center, is the nucleus. I use the phrase most of the time because in some cells of the body, the nuclei are actually found at the periphery. Okay, but in most cells, the nuclei are usually situated in the center. And most of the time, they are drawn or represented with a circular shape. Okay, I want everybody to understand that there's also another spherical structure that you can find inside the nucleus, and that is actually the nucleolus. Okay, and then I want everybody to understand the area between the cell membrane and the nucleus is called the cytoplasm. And I want everybody to take note that there are solid structures that you can find in the cytoplasm, and there are also fluid portions. You've learned from your basic science subjects or anatomy that we call these solid structures as organelles. Okay, they are named such because they are functional structures of the cell, so they act like organs in our body. So since they are smaller than the organs in our body, they are referred to as the organelles. The fluid where the organelles are suspended, we call it as the cytosol. I want everybody to take note. In an actual picture, okay, let's say for example, you are asked to view a specimen under the microscope, you're not supposed to expect that you will see all the structures that are presented in this photo. In reality, so this is how human cells would appear under the microscope, especially if we're using the bright field type or the compound type of microscope. That is the most commonly used type of microscope in histology laboratory. And I want everybody to appreciate that the first arrow in the PowerPoint slide is actually pointing to the cell membrane or plasma membrane of the human cell. The second red arrow is actually pointing to the cytoplasm. What do you notice? You cannot actually appreciate the presence of the organelles. Okay, what you only see is just an empty space. Okay, but you can always assume that there are organelles in there. And for us to see them, we need to use a more sophisticated and a more advanced type of microscope. The third arrow, is actually pointing to the nucleus, the largest organelle that we can see inside a human cell. That's why the size of the nucleus will actually make it easy for us to identify it in the under the microscope. And as you have learned from the previous slide, there's also another spherical structure that you can see inside the nucleus, and that is the nucleolus. So that's so I should expect that every time we will try to show you pictures of cells, you will be able to identify these four important structures. You have the plasma membrane or cell membrane, you have the cytoplasm, you have the nucleus, and you have the nucleolus. Okay? So again, cytoplasm is composed of both solid and fluid portions. The solid portions are referred to as the organelles, and the liquid portions are referred to as the cytosol. I want everybody to take a look at the photo. Number 1 is most likely the nucleolus. That's the spherical structure that you can see inside the nucleus. So number 2 is most likely the nucleus. And the solid structures that you can find outside the nucleus are referred to as the organelles, and the fluid where they are suspended is referred to as the cytosol. I want everybody to take a look at number 14. Number 14 is actually representing the border or the boundary of the cell, and that is the cell membrane or plasma membrane. So human cells have three basic parts. Okay, you have the cell membrane or plasma membrane, then you have the largest organelle, which is the nucleus with the nucleolus inside it, and you have the space between the two, the space between the cell membrane and the nucleus, and we call it as the cytoplasm with the organelles and cytosol. But according to some books, okay, it's not enough that we should say that cytoplasm is composed of both organelles and cytosol, because there is that third component to the cytoplasm, and we call them as the inclusions, or some books would refer to them as the inclusion bodies. So let's try to discuss the importance, okay, or the characteristics of these inclusion bodies. So just like the organelles, the inclusion bodies or inclusions are also solid. So that's why they are always compared to organelles. I want everybody to take note. Organelles are named such because they are functional components of the cell. Inclusion bodies, on the other hand, are not functional. So what's the importance of discussing inclusion bodies? I want everybody to take note that these inclusion bodies may or may not be present in the cell. Okay? I want everybody to take a look at this picture. As you can see, suddenly there's the appearance of two circles. So these two circles will actually represent inclusion bodies. Okay? At some point in the life of the cell, okay, these inclusion bodies might be present in its cytoplasm, okay, and at some point they might disappear. So why? Because majority of these inclusion bodies are considered to be as stored nutrients or stored waste products. Okay, so let's say, for example, if you are stressed with your academics and you tend to eat a lot of food, okay, then there will be buildup of fat in the body. Some of these fats, okay, will be deposited in your let's say hepatocytes or liver cells. Okay, so suddenly the cytoplasm of the liver cell will have a solid component that is not previously present on it, and that solid portion now will be referred to as inclusion or inclusion body. Okay, but if you decide to, let's say, start to lose weight because you think na you're already fat, okay, what will happen? As you are losing weight, okay, your body will be actually breaking down stored fats. Okay, so what will happen? The fat inclusion bodies, okay, stored in your hepatocytes will disappear. So that's why these inclusion bodies are not what you call this, considered as true organelles, because okay, they are not actually performing any function for the cell, and they are just representing stored nutrients or stored waste products. Okay, example of these inclusion bodies are lipofuscin, lipids, and glycogen, and we will discuss them one by one. So let me start with lipofuscin. I want everybody to take note that the organelle situated in the center is the lysosome, and you've learned from your anatomy and other science subjects that the lysosome is unique, okay, because it's the organelle that contains digestive enzymes. So most likely this organelle is involved in intracellular digestion. I want everybody to take note that the cells in the body can break down fats, lipids, and carbohydrates, and the digestion of these fats, lipids, and carbohydrates take place in our lysosome. But what I also want you all to take note, that the lysosome does not only digest carbohydrates, lipids, and fats. Let's say, for example, should there be presence of injury, and the injury cause some of the organelles inside the cell to become damaged, okay, and with the damage, these organelles are no longer functioning, so the cell, okay, will actually discard these organelles. Again, what will happen? These damaged organelles will be actually transported to lysosome, and the lysosome will digest the organelle. So what I want everybody to take note, as the lysosome is breaking down lipids, proteins, carbohydrates, and even damaged organelles, the accumulated waste products of the digestion will eventually become the lipofuscin. Okay, so that's the origin, okay, of our lipofuscin. So under the microscope, especially with H&E staining, lipofuscin pigment would appear to be a yellowish brown pigment. So that's why I intentionally typed in the word lipofuscin in brown color. And according to the PPT, this lipofuscin is actually composed of residues of lysosomal digestion. Okay, so I want you to imagine you have a cell in front of you. Okay, as the cell in front of you ages, okay, it would only mean that it has already metabolized a lot of substances. And as with the metabolism of these substances, the cell has already accumulated a lot of waste products. And you know very well that some of these waste products will become lipofuscin. So what does that mean? Okay, lipofuscin accumulates through the years. So therefore, lipofuscin can somehow give the pathologist or the one doing the examination of the histology slide the idea how old the cells that the person is looking at. Okay, so that's why the book, okay, would describe lipofuscin I mean, as the wear and tear pigment, because it actually represents the age of the cell. So that's why if you're going to compare cells A and B, you can say no that cell A is older than B because cell A has a higher amount of lipofuscin. But take note, if the cell with the lipofuscin will undergo mitosis, okay, the two daughter cells will no longer contain the lipofuscin. I tried my best to look for the reason for this one, but I cannot find any explanation in the book and even in the internet. So what I want you all to just, take note from this slide is that as the cell undergoes mitosis, the lipofuscin will actually disappear. So, aside from the fact that lipofuscin would indicate the age of the cell, okay, I want you all to take note that lipofuscin tends to accumulate in cells that are not actively undergoing mitosis. So, let's say, for example, you know very well that once we lose our neurons in stroke, okay, we cannot anymore replace them, okay? And you know very well that when you have an injury on your skin, okay, the fibroblasts in there and the keratinocytes are in there will actually multiply in number just to replace the damaged tissue. So the neuron, okay, has a limited capacity to multiply, and the keratinocytes and the fibroblasts in the skin have unlimited capacity to multiply. So if these cells will accumulate lipofuscin, okay, most likely expect that the fibroblasts and keratinocytes will lose their lipofuscin because they can undergo mitosis. While the neurons, since it cannot undergo mitosis, it has the chance to accumulate lipofuscin. So we can therefore conclude that lipofuscin is best demonstrated in organs or cells that are not anymore actively undergoing mitosis. And these are the examples of organs or tissues, okay, that are no longer active in mitosis. So therefore, these are the ones that are rich in lipofuscin. We have the nerves, okay, and let me be clear with this one. Old studies have shown that neurons cannot regenerate, but recent researches, okay, are suggesting that neurons can regenerate. It's just that the regeneration is too slow that they cannot replace the damaged neurons in time. So most likely a patient with stroke will die even before the neurons can regenerate themselves. But what's the point? Since neurons cannot undergo mitosis as fast as the other cells in the body, so therefore they can accumulate lipofuscin. Okay, same as with skeletal muscles and cardiac muscles. So that's why people who had acute myocardial infarction or heart attack are advised not to anymore engage in strenuous activities because, okay, they have already lost some of their cardiac muscles. Okay, if they will still engage in strenuous activities, then there's a chance that they will also be killing the remaining healthy cardiac muscles. So to explain the previous PowerPoint, I want everybody to take a look at these two photos of neurons that were taken from the brain. So I want you to locate first where are the nuclei, and I want you to appreciate inside the nuclei the presence of the nucleolus, but I want you to particularly focus on the cytoplasm. Okay? Can you appreciate the presence of yellowish brown pigment in the cytoplasm? Okay. So these are your lipofuscin pigment. So this is a picture of neurons taken from the peripheral nervous system. The brown pigment that you can see in their cytoplasm are lipofuscin. So these are the cardiac muscles. What do you notice? You've learned from your anatomy that cardiac muscles are striated. So it's very obvious in the picture. The muscle fibers that you can see in there are striated. But we will have a separate discussion on the other characteristics of the cardiac muscle. What I want you all to take note from this PPT is that in the cytoplasm of the muscle fibers of the cardiac muscles, you have their brown pigments, okay, which represent our lipofuscin. Okay, again, lipofuscin is best demonstrated in organs and cells that are no longer actively dividing. Let's have another picture of the cardiac muscle, and I want you to locate the presence of lipofuscin. So I'll give you 5 seconds, okay, to locate the lipofuscin pigment in the cytoplasm of these cardiac muscles. Next, so we will have another type of inclusion body. Okay, I want you to recall what you have learned from your teachers in biochem lecture, your favorite subject last semester. You've learned that as the glucose enters the cell, the glucose undergoes glycolysis in the cytoplasm. Okay, and the product of that glycolysis is 2-pyruvate molecule. Okay, and then the pyruvate will enter the mitochondria and it will be converted to acetyl coenzyme A. So just follow the arrow. I'm actually just discussing it step by step. Okay, then this acetyl coenzyme A will participate in the Krebs cycle. Okay, and the products of the Krebs cycle will proceed to the electron transport chain. So that's why you can see here na yung 6-NADH and 2-FADH2, okay, which are coming from the Krebs cycle, and these two will be transported to the inner mitochondrial membrane for the electron transport chain. Okay, this is how we convert, okay, glucose to ATP. But there are times in our lives that we tend to overeat. So what will happen? We supply our body with higher number of glucose, or with the amount of glucose that is above what is required by the body. But the body has a way of adapting to, increased level of glucose. Do you know what will happen? So let's proceed to the next slide. So in cases wherein there is excess glucose, okay, what will happen is the insulin will actually facilitate the storage of this glucose. So you see, the body has its way of adapting to the presence of excess glucose. So the body will not just allow that this glucose will be thrown into the urine and discarded. The body will actually store glucose. And I want you all to recall from your biochem that the storage form of glucose is glycogen. So that's why I want you to take a look at the picture of the muscle and the liver in the photo. Okay, what do you notice? The excess glucose will be transported by insulin to the muscle and the liver so that they can now be stored in the form of glycogen. So meaning, in a hepatocyte or the cell of the liver, you will suddenly see the appearance of an inclusion body, and that inclusion body now will represent the stored glucose in the form of glycogen. So let's discuss the characteristic of glycogen under the microscope. So glycogen is the storage form of glucose, and we have two important sites in the body where we usually store glycogen, and these are the skeletal muscles and the liver. The problem with glycogen is that it's not easily stained by H and E. So I hope you can still remember from the introduction to histology, hematoxylin imparts blue color to the nucleus, while the eosin will impart red color to the cytoplasm. The thing is, if this glycogen will appear as inclusion body in the cytoplasm of the liver cells or hepatocytes, this glycogen will not be stained by H and E. So they will not appear either as blue or red. Instead, they will appear as clear areas. So if you're going to take a look at the liver under the microscope, okay, you will appreciate the blue-colored nucleus, you will appreciate the red-colored cytoplasm, but within the cytoplasm, you can see clear areas. And these clear areas now will represent the glycogen. And just like what you've learned from the introduction to histology, there is that special stain that we can use for carbohydrates, and that's periodic acid shift. And it imparts red color to glycogen. I hope you will remember this last bullet in the PPT because we will apply that one on the succeeding slides. So let's try to take a look at the picture of hepatocytes under the microscope. So these are the hepatocytes, the active cells of the liver. Okay, what do you notice? I want you to focus on one hepatocyte. You can see the blue-colored nucleus, okay, that is stained by hematoxylin, and this nucleus is surrounded by red-colored cytoplasm. I want you to take a look at the cytoplasm. Can you see that some areas in the cytoplasm are not red in color? They actually appear as clear areas, and those clear areas now in the cytoplasm of these hepatocytes are the stored glycogen. And why do you think they appear as clear areas? Because they are not easily stained by H and E. So let's try to compare now the hepatocytes, okay, with someone who fasted overnight, okay, versus that to someone who had eaten 2 hours ago. Okay, so I want you to focus first on the picture on the left side. So these hepatocytes are from someone, okay, who fasted overnight. So as you can see, you have there the blue colored nuclei in the center of the cell, and it's surrounded by clear areas. So those clear areas are most likely representing the glycogen. Okay, so I want you to focus your attention on the picture on the right side of the screen. What do you notice? The clear areas suddenly became bigger, okay, and they are bigger, they appear bigger than the clear areas that you can see on the left side of the screen. Why? Because these hepatocytes, okay, the hepatocytes on the right side of the screen were taken from someone who had eaten 2 hours ago. So most likely this person had excess glucose in their blood, and the excess glucose were stored as glycogen in their liver. So that's why the clear area suddenly became or appeared bigger compared to the one who fasted overnight. So I hope I was able to explain that well, huh? Next. So I want you to take a look at these hepatocytes, okay, and they are actually surrounding a large hole in the center. That large hole kasi in anatomy and histology, we call that as the central vein. But you don't have to memorize it, ha? We have a separate discussion on the liver, okay, in the succeeding chapters in histology. But for now, I want you to just, memorize in your mind, how the hepatocytes are arranged around the central vein. Can you appreciate in the cytoplasm of the hepatocytes the presence of clear areas on those, and these clear areas are actually the glycogen. And the reason why they appear clear, okay, because they are not easily stained by H&E. And there's a special stain, that you can apply to demonstrate whether these clear areas are really glycogen and not fat. Okay, so what will you use again? Periodic acid shift, and the periodic acid shift will make these cells will make the glycogen appear red. Okay, so I want you to what you call this? Parang memorize. Okay, how the cells are arranged around the central vein. Okay, and let's have the same picture, but this time the cells are stained with periodic acid shift. So what will happen? Let's see. Can you appreciate that the cells, okay, the hepatocytes are still surrounding that central vein in there, okay? And suddenly, the clear areas that you saw from the previous slide appeared red in this photo. Why? Because this liver sample is stained by periodic acid shift, and periodic acid shift will make the glycogen inclusions appear red. So that will now confirm that the clear areas that we saw from the previous picture are really glycogen. Do you know that glycogen is also present in the cytoplasm of the cells of the lining epithelium of the vagina? Okay? But this time, what I want everybody to take note, the glycogen is present in the vagina not because we want to store glucose in the vagina. So if you're a guy, you do not talk to your girlfriend as if, ikaw na lang magtrabaho, oy, kay daghan man ka glycogen, napagani kay glycogen sa vagina. The glycogen in the vagina will serve another purpose, and it's not for storage of energy. Okay, I want you to focus at the two photos in the picture. Okay, both of these specimens are lined by stratified squamous epithelium. Letter A is actually the human skin. Okay, what do you notice? The cytoplasm is deeply red, okay, because the cytoplasm is stained with eosin. Compare the cut section of the skin to the photo in letter B. The letter B is actually the lining epithelium of the vagina. And focus on the cytoplasm of the cells. Can you see clear areas? Okay, so those clear areas actually represent the glycogen inclusion bodies in the lining epithelium of the vagina. So that's why whenever I see, okay, stratified squamous epithelium specimen in the histology laboratory, and once I see clear areas in their cytoplasm, I will know, okay, that the specimen that I am viewing is actually from the vagina. So take a look at this another photo of the vagina. Okay, you focus on its lining epithelium. It's stratified squamous, non-keratinized. And what do you notice? The cells in the lining epithelium of the vagina have clear areas within their cytoplasm representing the glycogen. So the next question is, what is now the purpose of storing or placing, okay, glycogen in the cytoplasm of the cells of the vagina? take note that the glycogen present in the cytoplasm of the cells of the lining epithelium of the vagina is actually acted upon by the bacteria present in the vagina. These bacteria are the normal flora of the vagina, meaning they are normally present in there. And we call them as Lactobacillus acidophilus. The name of the spelling of the name of the bacteria is indicated on the lower left side of the screen. Okay, and this Lactobacillus acidophilus will convert the glycogen, okay, into lactic acid, thereby making the environment inside the vagina acidic, so that the acidity will not promote the growth of microorganisms, thereby protecting the vagina from infection. Okay, so that's also the reason behind why semen has to be alkaline, or else the sperm cells will be killed by the acid, the lactic acid in the vagina. So that's why we have this product, no, that we can actually buy from the market, and it's called Lactacid. Lactacid, it's a female vaginal wash because it's supposed to maintain the acidity in the vagina to prevent infection. So you see, I hope you have realized that the glycogen in the vagina is not stored form. It's not for the purpose of storing glucose for future energy use. Okay, it's actually for the formation of lactic acid. Another inclusion body is actually hemosiderin, and intentionally typed down the word hemosiderin in brown color because this one also appears brown, okay, or it appears as a brownish pigment. Unlike lipofuscin, hemosiderin is actually the breakdown product of hemoglobin. So in areas in the body where red blood cells are destroyed, and you know very well that inside the red blood cells we have hemoglobin, so if these red blood cells are destroyed, so the hemoglobin will also be degraded, and once the hemoglobin will be degraded, the end product will be the formation of the brown pigment, and we will call it hemosiderin. Okay, so where can we find hemosiderin? In areas in the body where there is hemorrhage or bleeding, and I will explain later on why a hemorrhage or a bleeding will eventually lead to the formation of hemosiderin. Remember, hemosiderin is a breakdown product of hemoglobin. Second bullet in the PPT would say this hemosiderin is normally present in the red pulp of the spleen. Okay, I hope your anatomy teachers have taught you that the graveyard for red cells, okay, is actually the spleen. So if red blood cells are damaged, if red blood cells are already more than 120 days old, okay, they are actually destroyed in the spleen. So again, if they are destroyed in the spleen, particularly in the red pulp of the spleen, you expect that hemoglobin will also be degraded there, okay? And then what will happen? The red cell will be degraded, the red cell will be destroyed, the hemoglobin will be degraded, and hemosiderin will be formed, okay? And according to the PPT, alveolar macrophage will also demonstrate hemosiderin when there is bleeding to the alveolar space, and I will explain that later on. Let's start with the spleen, okay? So this is the cut section of the spleen, and you've learned from anatomy that it's composed of white pulp and red pulp, okay? The white pulp is actually rich in lymphocytes or white blood cells, so that's the reason why it's named white, okay? And I want you to appreciate the white pulps appear to be nodular or rounded in shape, and the white pulps are surrounded by the red colored areas, and we refer to them as the red pulp. So that's where we destroy our old and damaged red blood cells. So this is how the spleen will look like under the microscope. So the nodular areas there are most likely representing the white pulp, okay? And the red arrow is now pointing to the area we refer to as the red pulp, okay? So it is in the red pulp where red blood cells are destroyed. So expect it is also in the red pulp that you can see hemosiderin, okay? So again, the nodular patterns that we see in the spleen are the white pulps, okay? And then I want you to focus on the right side of the screen, okay? Can you see the ones labeled as splenic sinuses, okay? So those are spaces within the red pulp where the red blood cells will actually flow, okay? And these sinuses, okay, are too small that red blood cells have to be flexible in order for them to squeeze themselves into these sinuses. If the red blood cell is damaged, if the red blood cell is, let's say, old, okay, it has already lost its flexibility. So what will happen? The red blood cell will most likely be trapped or stranded in these sinuses. And when they are trapped or stranded, they will be eaten by macrophages, okay, which are present on the splenic cords. Okay, so the splenic cords are composed of macrophages, and they are just waiting for red blood cells to be trapped in the splenic sinuses. And when they are trapped, okay, they will be eaten up. Okay, and since the red blood cells are eaten up by the macrophage, they will be destroyed, hemoglobin will be degraded, and you will now have the formation of hemosiderin. Can you appreciate the presence of a brown colored cell in the photo? Okay, so most likely that's a macrophage, and it has previously destroyed or captured a red blood cell from the splenic sinus. Okay, so these are also pictures of the red pulp. Okay, I want you to take a look at the splenic sinuses. Okay, and in the first photo, okay, the one labeled as macrophage appears to have a brown cytoplasm. So most likely that brown pigment is hemosiderin, and it would signify that that macrophage, okay, has already digested, okay, or destroyed a red blood cell. The blue arrow on the second photo, okay, is actually also pointing to a macrophage with a brown colored cytoplasm. So that brown colored pigment in the cytoplasm of that macrophage is again hemosiderin. Okay, I hope I was able to prove to you that this hemosiderin is normally present in the red pulp of the spleen. But hemosiderin can also be present in areas where there is hemorrhage or bleeding. So I'll give you an example. So I want you to take a look at the lungs, okay? And you know very well that the terminal portion of the lungs, okay, are called the alveoli. And I want you to take a look at the picture of the alveoli. They actually look like yung clusters of grapefruit, right? Okay. And I want you to take note that once we cut the alveoli, you do not expect them to appear like grapes, but instead they would appear to be as you can see them in the photo, they would appear to be groups of circles, okay, positioned beside each other. So that's why, take a look at the next photo. This is how now the alveoli will appear under the microscope. So just imagine yung grape-like clusters, okay, they are composed of circular structures, okay, found beside each other. So most likely the circular structures here are representing the alveolus, okay, or the alveolus that are grouped together in a grape-like clusters, right? Okay, so this is how the alveoli would appear under the microscope. In some people with, let's say, for example, Mycobacterium tuberculosis, okay, these people would have destruction of the walls of their alveoli. And if the walls of the alveoli will be destroyed, then most likely there would be bleeding. So that's why what you notice in the spaces, okay, inside the alveoli, you have now there the presence of red blood cells. And the presence of these red blood cells will most likely affect gas exchange. And the person with this type of condition will most likely have difficulty of breathing. So there's a need for the body to remove the red blood cells from the alveolar space to restore the normal gas exchange, okay, function of the lung. So I want you to proceed to the next slide. So I want you to take a look at the photo on the left side of the screen. As you can see, the photo is showing you normal alveoli, okay? And take note of the presence of the alveolar macrophages in the alveolar space, okay? So if these alveolar spaces will be filled up with red blood cells, okay, these macrophages will try to clear them by ingesting them, okay? So I want you to focus on the photo on the right side of the screen. The yellow colored cell in there is actually a macrophage, and as you can see, it's surrounded by red colored cells. So just assume that this macrophage is present in the alveolar space and there's bleeding within the alveolar space. Can you appreciate yung sa blue arrow that the cytoplasm, okay, or the cytoplasmic process of the macrophage has already enveloped or engulfed a red blood cell. And you know what will happen? This red blood cell will eventually be eaten up by the macrophage, broken down, and the hemoglobin will be converted to hemosiderin. So I want you to take a look at this cut section of the lung. So I also want you to take note of the presence of the brown pigments in the cytoplasm of the alveolar macrophages shown in the photo. So this one might be, diagnosed as a mild hemorrhage. So just try to imagine if the specimen that the pathologist will view will look like the one in the next slide. So in this photo, you can see a lot of alveolar macrophages with brown pigments in their cytoplasm. So this will give the pathologist the idea that previously there was a massive bleeding or hemorrhage in this alveolar space. And this brown pigment again is what? Hemosiderin. So let's have a review. There are already two brown pigments you have learned so far, Lipofuscin and Hemosiderin. Lipofuscin is normally present in cells that are no longer active in mitosis, okay? While Hemosiderin is present in areas in the body where there is what? Hemorrhage, or where there is destruction of red blood cell, or breaking down of hemoglobin. And the next inclusion body that we will discuss is lipid. And since our subject is histology, we're supposed to discuss normal structures and normal functions in the body. So we will not discuss yung deposition of fats in the walls of the artery, deposition of fats in the liver. So we'll just focus on the normal areas in the body where we usually deposit, or we see deposits of fat. And the most common site, okay, for these fats to be deposited is the adipose tissue, or the adipose cells, okay? So I want you to take note, these lipids, okay, will accumulate through time. And as it accumulates, okay, it has the tendency to push the organelle and the nucleus of the adipocyte or adipose cell to the periphery, okay? So I want you to take a look at the cells in the photo on the right side of the screen, okay? The cells that appear to have clear cytoplasm, okay, actually are adipose cells, and the clear portions in the cytoplasm are actually the fats that they have accumulated through time, okay? And I want you to take note that even if these spaces appear empty, okay, they previously had fats inside them. It's just that when we did the tissue processing, especially when we did the clearing and the dehydration step, the alcohol and the clearing will actually dissolve the fat, leaving behind an empty space or a hole. But you can always assume that the hole was previously occupied by fat, okay? So let's say, for example, you're having difficulty in parasitology, and your teacher in parasitology lab will give you a practical exam, a one-on-one exam in using the Zoom account. So what will happen? You will be stressed, and you will start doing yung stress eating. And as you do the stress eating, there will be accumulation of fat now in your adipocytes. And what will happen? Since your teacher loves giving you moving exam or practical exam every meeting, so what will happen? You will have several episodes of stress eating, and the fat will continue to accumulate in the cytoplasm of your adipose cells. And what do you notice? As the fat becomes bigger, or the fat inclusion body becomes bigger, the nucleus is somehow displaced towards the periphery, okay? So let's try to look at the progress of this enlargement of the fat inclusion body. So again, the fat is steadily increasing while the nucleus is displaced towards the periphery, until such time, okay, that the fat will become so big that it has already displaced the organelles and the nucleus in the periphery, and it has also caused the nucleus to become flattened, okay? So let's apply that one on an actual slide. So I want you to take a look at these adipose cells, okay? I want you to take a look at their nuclei, okay? Majority of these cells have flattened nuclei, and their nuclei are not found in the center of the cell, okay? Instead, the nuclei are found at the periphery, okay? Because they were displaced by the presence of the fats, or fat inclusion body, okay? So this is again another picture of showing you adipose cells. So I want you to appreciate the presence of the fats in the center with the peripheral flattened nuclei. The next inclusion body or pigment that we will discuss is melanin. And I know you know, okay, that melanin is the pigment responsible for the brown color of the skin. So that's why the picture that you see in there is lined by stratified squamous keratinized epithelium. But what I want you all to focus at the base of the epithelium. Can you see the presence of brown pigment? Okay, so those brown pigments now are the melanin pigments. So I hope you can still remember Lipofuscin, okay, and Hemosiderin are both brown in color. So it's also very important that you should be able to differentiate Lipofuscin and Hemosiderin from melanin. But melanin is easy to identify as long as you know it's the skin, as long as you see that there's stratified squamous keratinized epithelium. Anything brown pigment, anything brown color associated with it will most likely be melanin. Okay, so let's try another picture, huh? So can you appreciate that there's a stratified squamous keratinized epithelium in the photo? Yes or no? Yes, so most likely you have an idea that this specimen is indeed the skin. Okay, can you appreciate the presence of brown pigment on the base of the lining epithelium? So most likely you know that those brown pigments are the melanin pigments. So I want you all to review, okay, where can you find Lipofuscin, where can you find Hemosiderin, where you can find melanin, because I might be using photos in our quizzes and I will be asking you to identify the inclusion body. So, as we discuss the cell, we should start with one of its components, okay, and that's the cell membrane. And you've learned from the discussion a while ago that the cell membrane is the outermost boundary of the cell. It's actually the border of the cell, and what regulates the entry and exit of substances in and out of the cell is actually the cell membrane. The most important thing I want you to take note from this PowerPoint slide is that the cell membrane of humans, anyways, that is true to almost all organisms. The cell membranes are actually referred to as bilipid layer. Okay, what do you mean by the word bilipid? Two layers of lipid. I want you to take a look at the white arrow. The white arrow is actually pointing on one layer of the cell membrane. Okay, and the yellow arrow is pointing to another layer of the cell membrane. Okay. So therefore, this white arrow and yellow arrows are proving to us that the cell membrane is actually composed of two layers. And majority of the components of these layers are lipids. So therefore, we can conclude that the cell membrane is composed of two layers of lipids. So this photo is a good representation of the human cell membrane. Okay, what do you notice? You have there two layers of fats. That's why the cell membrane is described to be bilipid. Okay, what we will do on this slide is to identify the components of the cell membrane. Okay, so let's start with the first one. Okay, the blue arrow is pointing to the phospholipids, okay, the ones that look like balloons. Okay, and from the picture you can conclude that the most abundant component of the cell membrane is the phospholipid, the one pointed by the blue arrow. The next component, okay, is the yellow colored structure inserted among the phospholipids. Okay, and that yellow colored structure is actually cholesterol. Again, blue arrow phospholipid, yellow arrow cholesterol. Okay, the pink colored structure that looks like safeguard soap, okay, is actually the protein. Okay, so let's start from the very beginning. Blue phospholipid, yellow cholesterol, pink protein. The component of the cell membrane that is always displayed outside, okay, the one pointed by the green arrow, is the carbohydrate. So, we therefore have four components of our cell membrane. We have the phospholipid, okay, which is the most abundant component of the cell membrane. Inserted among the phospholipid, you have there the cholesterol, okay? And the large appearing structures of the cell membrane, we call them as the proteins. And the structure that is always displayed outside the cell membrane, you have there the carbohydrates. So we will be discussing these components one by one. So let's start with the most abundant. So the phospholipids make up the majority of the bilipid layer of the cell membrane. So I want you to take a look at the highlighted photo of the phospholipid. Okay, as you can see, it has a head. It's actually labeled for you guys. The rounded portion of the phospholipid is called the head. Okay, and you have there two tails. The head of the phospholipid is actually composed of phosphate, and phosphate is highly hydrophilic. So therefore, it will interact with water. So that's the one encircled in the photo. That's the head. That's the hydrophilic portion of the phospholipid. On the other hand, the two tails of the phospholipid are composed of fatty acids. And from the name itself, you can clearly predict that this portion of the phospholipid is highly hydrophobic. So therefore, phospholipid has both hydrophilic and hydrophobic portions. And if you can remember from your biochem, if a substance has both hydrophilic and hydrophobic portions, we describe the substance as amphipathic. Amphipathic. Okay, so I hope you will take note of that word because I might ask that in the exam or quiz. Okay, so let's have a review. The heads, okay, the phosphate heads of the phospholipids are hydrophilic, okay, while the tails, which are composed of fatty acids, are hydrophobic, okay. And when you say hydrophobic, the term that would best represent it is non-polar. When you say hydrophilic, we can describe it as polar. So given that we have two layers of phospholipids in the cell membrane, because cell membrane is bilipid layer, the next question is, how are the phospholipids arranged in the bilipid layer? So that will be best answered by the next slide. Since there's water outside the cell and the cytosol, okay, within the cell, the cytoplasm within the cell is also rich in water, thereby, okay, the phospholipids are arranged in such a way, okay, that the hydrophilic portions are the ones facing the water. So that's why what you notice in the picture, the phosphate heads of the phospholipids are directed towards water, okay, while the fatty acids, since they are hydrophobic, they are actually situated in the center and they are hidden from the water. Do you know, that phospholipids tend to migrate from one place to the other? So laagan din ni mga phospholipids bisag walay kwarta, kay walay allowance, kay pandemic, kay online class naman, no? So let's move to the next slide. Since we have learned that phospholipids tend to migrate from one place to the other, so let's try to see how will it affect, no, the other components of the cell membrane. So I want you to focus on the photo. You have to focus first on the yellow colored proteins or receptors that you can see on the cell membrane. These yellow colored receptors are the receptors to insulin, okay? So whenever insulin is present, okay, this insulin will bind to the proteins, okay? And these proteins will respond by what? Causing, okay, the blue colored glucose transporter to open. And if it is open, the glucose molecules now are free to enter the cell. So that's why it's a common knowledge that glucose, okay, will never enter the human cell without the presence of insulin. If insulin is absent, glucose will remain in the blood, and that will now cause hyperglycemia, and the patient might be diagnosed to be diabetic. Okay, I hope it makes sense, huh? Okay. So, next, let's move to the next slide. We have here the bilipid layer of the membrane composed of phospholipids which tend to migrate, okay, and you have here now the glucose transporter, okay, and with the action of the insulin, this glucose transporter will open up, allowing glucose to enter the cell, okay. So you now have glucose molecule in the cytoplasm that will undergo glycolysis so that it will be converted to pyruvate later on, and the rest will follow, yung Krebs cycle and electron transport chain. The problem is, okay, so what will happen if the membrane is highly fluid, meaning the phospholipids are allowed to migrate, no, to different sites in the cell membrane? There's a tendency that the phospholipids will also drag along with them the components that are found beside them. Okay, so they can change the location of the other components of the cell membrane. So what will happen if they will change the position of the glucose transporter? The next time glucose molecule will arrive, okay, it has no glucose transporter to enter to. So thereby, there's a probability that the glucose will stay in the blood and it will cause hyperglycemia. So, what will now keep this glucose transporter in place so that once glucose is around, glucose can freely enter it? Okay? That is now where the role of the cholesterol will become very important. Okay? So I want you to appreciate the phospholipids on the photo on the right side of the screen. Okay? They have these blue colored phosphate heads and you have there the black colored fatty acid tail. Okay? Can you appreciate the yellow colored cholesterol molecules that are inserted among these phospholipids? Okay, do you know the role of the reason why this cholesterol has to be inserted between them? The cholesterol will actually restrict their movement. So if these phospholipids will stay in place, the glucose transporter will also stay in place. So there's a reason why we need to have cholesterol inserted among phospholipids so that the movement of these phospholipids will be limited. Okay, so I want you to focus on the yellow colored cholesterol. Let's discuss its parts. Okay, I want you to take a look at this one. So that's the hydrophobic ring of your cholesterol. I know you have already learned this one in your biochem, no? You call it as per-hydrocyclopentanophenoline ring, okay? But in histology, it's enough that you know it's the ring of the cholesterol and it's hydrophobic, okay? And then next, the hydroxyl group that you can find on the terminal portion of the cholesterol is hydrophilic. So therefore, your cholesterol, okay, has both hydrophilic and hydrophobic portion. The hydrophobic being the ring, okay, and the hydrophilic representing the hydroxyl group. So therefore, this cholesterol behaves like that of your phospholipid and they are both amphipathic. Okay, I hope na-explain ko yun ng well, ha. So, yeah. So we'll move to the next slide. So I want you to take a look at the cholesterol in the picture, okay? The yellow circle in the cholesterol is representing the OH, the hydroxyl group, okay? And then you have there the 4-fused ring, the per-hydrocyclopentano ring. So if you will be asked in the exam, how is cholesterol arranged in the cell membrane, okay? You have to answer that the hydroxyl group, since it is hydrophilic, okay, it is located at the area where you can find the phosphate heads of the phospholipids, which are also hydrophilic, while mixed along with the hydrophobic fatty acid tail is the 4-fused ring or the per-hydrocyclopentanophenoline ring of the cholesterol. Okay? So what's the reason why? What's the reason behind why cholesterol has to be present in there? To restrict the movement of the components of the cell membrane. So you have here the two layers of the cell membrane composed primarily of phospholipids. You have the glucose transporter, okay? And then you have the addition of cholesterol. And this cholesterol will restrict the movements of the phospholipids, thereby keeping the glucose transporter in place so that the glucose can freely enter it whenever there's the action of insulin. Okay? So there's that certain portion in the cell membrane that we refer to as the lipid raft. Okay? So you have there a photo and there's this certain area in the photo that is labeled lipid raft. What do you notice? You have there the abundance of the blue colored structures. Okay, I want you to take note that these blue colored structures are the protein components of your cell membrane. So what can you say? The lipid raft, okay, is abundant in proteins. The next question is, if you have there the abundance of proteins in the lipid raft, why call it lipid raft and not protein raft? Okay, I want you to take a look at the lipid raft. Can you see the yellow-green colored structures that you see inserted among the proteins? Okay, those yellow-green colored structures are the cholesterol molecules. So the reason why these proteins are somehow kept in place and they are aggregated on one particular area of the cell membrane, it's because they are not moving apart from each other, because they are restricted, and what restricts them is the presence of cholesterol. And since there's abundance of cholesterol in the area was then referred to as the lipid raft. So let's have a review. These are your phospholipids with the phosphate head and fatty acid tails. Okay, and this is your protein. Okay, as you can see, it's aggregated along with other pink colored or fuchsia colored proteins in the picture. Okay, then you have here your carbohydrate. Okay, and then you have there the green colored cholesterol. Okay, what do you notice? The fuchsia colored and the pink colored proteins are aggregated on one particular area of the cell membrane, and that particular area of the cell membrane is rich in cholesterol. And what is keeping these proteins in place, okay, so that they can interact with each other, is the abundance of cholesterol. Okay, I hope it makes sense, ha? Sige, let's proceed to the next slide. You know very well that there are chemicals, okay, that can make cells react by binding to the receptor. So you can clearly see in the animation that the chemical, okay, has bound to the receptor. And what do you notice? The receptor, yung parang may mga lumpia, okay, is actually composed of several proteins, okay? And these proteins are aggregated on one area of the cell so that they can form one receptor. Okay, so this is one of the importance of the lipid raft. Okay, for a receptor to be formed, okay, the proteins must be close to each other and they should not be moving apart from each other. Okay, so I want you to also to appreciate that the receptor also interacted with a protein that is located within the vicinity of the lipid raft. Okay, so for the main receptor to activate the yellow-green colored protein and for the cell to react, okay, this receptor and the yellow-green protein should be concentrated on one area of the cell. And that's the purpose now of having a lipid raft. Okay, so to best explain this one, let's move to the next slide. Please listen first to my explanation before you mind the animation. You have there the lavender colored chemicals that are supposed to stimulate this cell. Okay, so as you can see, the lavender colored molecule activated the receptor, which is composed of group of proteins, by the way. Okay, and what do you notice? The red colored receptor activated the blue colored protein. Okay, and the blue colored protein activated other proteins that you can see inside the cell, so thereby the cell now will react to the presence of the lavender colored chemical present outside it. Okay, so just try to imagine if lipid raft is not present, or if cholesterol is not inserted among phospholipids, then the red colored receptor and the blue colored protein, okay, will not be kept in place, so thereby they cannot interact. So even if the lavender colored molecules are present outside the cell will not respond to their presence. So again, I want you to take a look at the G-coupled receptor that is composed of 7 proteins. Okay, so just try to imagine if there's no cholesterol inserted among them, most likely these proteins will migrate apart from each other. And you can also see that the G-coupled receptor is interacting with yung alpha, beta, and gamma subunits. So just refer to this blue arrow. Okay? So as soon as the ligand, yung ano bang color yan? Violet colored ligand will interact with the G-coupled receptor. Okay? So just follow the black arrow. The G-coupled receptor now will activate the alpha, beta, and gamma subunits. And this signal transduction, okay, was made faster by the fact that the proteins, okay, are found almost at the same area. And why do you think they are found in the same areas? Because they are not moving from each other. What's the main reason behind it again? Because they are their movements is limited by cholesterol. So you see, there's also the importance of having cholesterol in the cell membrane. So the third component of cell membrane is the protein. Okay. Again, the proteins in the cell membrane can be divided into integral and peripheral. The integral is also known as transmembrane. And according to the PPT, okay, it's directly incorporated into the cell membrane and it can act as channels, carriers, transporters, and even as receptors. Okay. The second type of proteins are the ones that are loosely associated with one of the membranes. That's why they are easily extracted from the membrane. Okay. And take note, okay, these proteins, they may not be able to act as transporters, channels, carriers, but they can be responsible for the maintenance of the shape of the cell. And to best explain that one, so I created this animation. Do not judge my my animation or yung effects sa slides because I'm not really good at it. So I want you to imagine a cell, okay? So there's a cell in front of you. The two red lines that you see on the left side of the screen are representing the cell membrane on the left side of the cell. Okay. The two red lines on the right side of the screen are representing the cell membrane on the right side of the cell. So basically, you're looking at a single cell. Okay. The blue circles in the photo are representing peripheral proteins. Okay, because these circles are only interacting with one of the layers of the cell membrane. Do you know, okay, that these peripheral proteins actually interact with each other and they tend to pull each other, okay? And as they pull each other, they also cause the cell membrane to be pulled, okay, towards the center. So that's basically the reason why red blood cells are described to have a biconcave shape. And the depression that you see in the photo is created by the interaction of the peripheral proteins present in the cell membrane of the red blood cell. So therefore, we can conclude, okay, that peripheral proteins can contribute to the shape of the cell. Okay? So take a look at this photo. Okay? You have there the proteins labeled as ankyrin, and you have there another protein labeled as actin. So these are peripheral proteins of the cell membrane of the red cell. In addition to ankyrin and actin, there's also another peripheral protein, okay, that will make the ankyrin and actin interact with each other, and that is the spectrin. So just try to imagine the ankyrin and actin will actually pull each other using the spectrin, thereby causing now the biconcave shape of the red cell. Do you know that there's that certain medical condition we call as the hereditary spherocytosis, and these people are born with no gene for spectrin? So just try to imagine, if spectrin is absent, ankyrin and actin cannot anymore interact with each other, thereby, okay, they cannot anymore maintain the biconcave shape or the donut shape of red blood cells. So that's why the red blood cells will appear to be spherical in shape. Okay? And I want you all to imagine the biconcave shape helps the red blood cells squeeze through small capillaries. Okay? And for a red cell that is spherical in shape, it would be hard for it to squeeze itself into smaller capillaries. So what will happen? The red blood cells of people with hereditary spherocytosis will be destroyed in the circulation. They will rupture because of the pressure, because the cells behind them will be pushing them towards smaller areas in the capillaries. So that's why patients born with hereditary spherocytosis will develop anemia. Freeze fracture model was actually developed to study particularly the integral proteins present in the cell membrane. Okay, so how do we perform freeze fracture method in the laboratory? We place cells or we expose cells to freezing temperature. So we will allow them to freeze. Okay, then after these cells are frozen, okay, we will strike a force using a hammer. Okay, and you know what will happen? The force will cause the membrane to become fractured. And most of the time, the fracture is created, okay, in the hydrophobic portion of the cell membrane. And when you say hydrophobic portion of the cell membrane, that is the area where you can find the fatty acids, no? The fatty acid portions of the phospholipid. So if that's the case, the membrane will be separated into two layers, okay? So what you can see in the photo, the membrane is separated into two. One layer is called extracellular layer because it's found on the outer part of the cell. The other layer is called cytoplasmic layer because it's the portion of the membrane that is closer to the cytoplasm, okay? And take note, these cell membranes will now be viewed under electron microscope. So you have to take note of that. I did not put that one in the PPT. Freeze fracture method would require that the cell should be viewed under electron microscope. And you know what scientists found out when they tried to study the cell membrane? Okay, they noticed that when they examine, just look at the results, the portion in the right side of the screen, when they examine the extracellular layer of the membrane and as well as the intracellular or the cytoplasmic layer of the cell membrane, they saw protrusions. Okay, and you know very well that these protrusions are actually caused by the presence of the integral proteins. And do you know what they notice also? For every protrusion on the extracellular layer of the membrane, there's a corresponding depression on the cytoplasmic layer of the cell membrane and vice versa. So thereby, using freeze fracture method, they were able to prove, okay, the presence of the integral or transmembrane proteins. So this is how the extracellular layer of the cell membrane. So this is just one of the layers of the cell membrane under electron microscope. Can you appreciate the presence of protrusions? So those protrusions are actually caused or produced by the presence of the integral proteins. The last but not the least among the components of the cell membrane are the carbohydrates. And I hope you can still remember that I told you this is the component of the cell membrane that is always found outside the cell. So its function has something to do with the interaction of the cell to its environment. Okay? And take note, these carbohydrates can be referred to as glycolipids and glycoproteins. I know you know very well that the term glyco would refer to sugars or carbohydrates, right? Okay, so these carbohydrates can be glycolipids and glycoproteins. I want you to focus yourself on the red box, okay, or the red rectangle. I want you to appreciate the presence of the carbohydrate on the outer portion of the cell, and you can clearly see that this carbohydrate is attached to the phospholipid, which is a lipid. So you have a carbohydrate attached to a lipid, glycolipid. Okay? Focus on the blue rectangle. Okay? The blue rectangle is also pointing to the carbohydrates present on the outer portion of the cell membrane. But this carbohydrate is attached to a protein. So a sugar attached to a protein, glycoprotein. Okay? Take note that these carbohydrates, may it be a glycolipid or a glycoprotein, okay, are used by cells for, number one, cell adhesion, mupilit, mudikit, okay, and cell-to-cell interaction. So, I will be explaining you or giving you some situations or clinical scenarios wherein this cell adhesion, wherein these carbohydrates will be used for cell adhesion or cell-to-cell interaction. So this is a blood vessel and you can see the blood cells flowing through its lumen, okay? Normally, majority of the white blood cells are circulating in the central portion of the blood, okay? While majority of the red blood cells are circulating at the periphery. Okay, why? Because these red blood cells have to share oxygen to the cells and they have to pick up carbon dioxide. And for their function not to be interfered, majority, I'm not saying all, majority of the white blood cells should be positioned in the central portion of the blood. The yellow colored cells there in the picture are representing our white blood cells. So you can clearly see in the photo that they are positioned most of the time in the central portion of the flowing blood. Should there be any infection, these white blood cells have to go to the margin or border of the blood vessel, and they have to go out from the blood vessel and go to the area in the body where there is an ongoing infection. So just try to imagine if you're the white blood cell, okay, effort na siya muadto sa side, okay, para makagawa sa artery. Why? Because you have to do it while the rest of the cells behind you are also pushing you forward. Okay? So what do you think the cell, this white blood cell, would do in order for it to successfully go out of the blood vessel? So just try to imagine you have a white blood cell in the central portion of the flowing blood, and this white blood cell has to go to the periphery, okay, in order for it to reach the wall of the blood vessel and go out of the blood vessel, given that the blood will still continuously flow and the cells behind the white blood cell will push it forward. So how do you think white blood cells, okay, can still successfully go out of the blood vessel? So we'll discuss that one on the next slide. So I want you to take a look at this photo. You have here on the lower part of the photo, you have the endothelial cells. These are the lining epithelium of your blood vessel. So most likely these endothelial cells are representing the wall of the blood vessel, okay? And you have here a neutrophil or a polymorphonuclear leukocytes, okay? And this polymorphonuclear leukocyte has to go from the central portion of the blood towards the periphery so that it can go out of the blood vessel. And how do you think this neutrophil will be able to do that? Okay, can you appreciate the presence of the green colored structure surrounding the cell membrane of the neutrophil? Okay, and can you appreciate that the neutrophil is using this green colored structure to attach itself to the structures displayed by the wall of the blood vessel? Okay, these green colored structures helped the white blood cell, okay, to attach itself to the wall so that it can easily go out of the blood vessel, okay? And these green colored structures are the carbohydrates, the glycolipids, and the glycoproteins. So that's why we can conclude, okay, that carbohydrates, glycolipids, and glycoproteins can help or mediate cell adhesion, okay? So this is how the white blood cells will use the carbohydrates on its surface to attach itself to the wall of the blood vessel so that it will not be pushed back to the center by the cells behind it, okay? You can see that with the help of the carbohydrates, the white blood cell was able to go out of the blood vessel and phagocytose the invading microorganism. And if you can remember, according to the PPT, carbohydrates, aside from functioning for cell adhesion, they can also function for cell-to-cell interaction. So I will be giving you an example so that it would be easy for you to memorize. So as you can see here, you have a pathogen, okay, an invading microorganism, and this invading microorganism was eaten up by the macrophage. And can you appreciate in the animation that certain portions of the pathogen, okay, were displayed on the surface of the macrophage, okay? See, the structure that was used by the macrophage to display that the small parts of the pathogen is actually a carbohydrate, okay? So I want you to take a look at this picture. You have here 4 green colored bacteria, okay? And out from the 4, one was eaten, okay, by the macrophage, okay? And you can clearly see in the photo that the bacterium was eventually digested by the lysosome of the macrophage, and you can see that a certain portion of the bacterium was loaded, okay, into a certain structure, the blue colored structure, and together with the blue colored structure, the portion of that bacterium was displayed on the surface of the macrophage. And that blue colored structure now is primarily composed of carbohydrate, okay? So as you can see in the photo, out of 4 bacterium, 1 was killed, and you still have 3 bacteria to deal with. So you have here a microbe that is phagocytosed by this macrophage. You can clearly see that the microbe was broken down or digested in the cytoplasm of the macrophage, and the certain portion of the macrophage is loaded into this green colored structure. And take note, this green colored structure contains carbohydrates. carbohydrates. Okay, and this one, the carbohydrate containing structure and the portion of the microbe is together displayed on the surface of the macrophage. And do you know what will happen after? This macrophage will interact with T helper cell because it will present that portion of the microbe to T helper cell. And the T helper cell will also respond by using a receptor which can also contain carbohydrate. So therefore, this picture will prove to you, okay, that carbohydrates can mediate cell-to-cell interaction. The T cell that was activated by the macrophage using the carbohydrate, okay, will interact with B cell. And do you know what will happen? This B cell will eventually transform to form what we call plasma cell, okay? And this plasma cell will produce antibodies. I hope you can still remember that only 1 out of the 4 bacteria was killed by the macrophage, okay? And you know what will kill the other 3 bacteria? The antibodies produced by the plasma cell. So you see, that's how the immune system actually works, okay? So it would only take 1 to cause harm to the other bacteria. So 1 was sampled by the macrophage, and using the carbohydrate was presented to T helper. T helper activates B cell. B cell becomes plasma cell, and plasma cell will produce antibodies that will kill the other related bacteria. I will not require you to memorize T cell, B cell, macrophage, okay? I am just actually showing you, okay, how a carbohydrate will help cells interact with each other. So therefore, we can conclude carbohydrates can mediate cell adhesion, and the best example is the chemotaxis, and number 2, cell-to-cell interaction. Since one of the functions of the cell membrane is to regulate the entry and exit of substances in and out of the cell, it's also important to discuss how these substances are actually transported. Okay, the first type is called passive transport. So this photo is actually showing you how a passive transport is done. It's actually caused by the movement of molecules from higher concentration to lower concentration. Okay, it's very obvious in the picture. Just follow the direction of the red arrows. It's showing you that the molecules are moving from area of higher concentration to lower concentration. And there are 2 types of passive transport, simple and facilitated. Okay, the first one that you can see in the photo, wherein the molecule will just simply diffuse across the membrane without the help of a channel or a carrier or transporter, okay, that is called a simple passive transport. Okay, but if the movement of the molecule is from higher concentration to lower concentration, but there should be the presence of a channel or a carrier, that type of passive transport is called facilitated. Okay, and since molecules behave this way, behave in a manner na they will move from higher concentration to lower concentration, the cell will not have to spend energy for these molecules to move because these molecules will move on their own. So passive transport does not need energy. Okay? So again, simple diffusion is the movement of molecule from higher concentration to lower concentration directly across the cell membrane. So if the movement of the molecule from higher concentration to an area of lower concentration would require the presence of a channel or carrier, it's called a facilitated type of passive transport. So I want you to take a look at the lavender-shaped proteins in the picture. These are actually called channels. So there's a difference between a channel and a carrier. So let's try to talk about the channel first. You can see that these channels are allowing the transport of the red circles, okay, from an area of higher concentration to lower concentration. If the protein does not change its shape, okay, as the molecule is transported, it's called a channel. So how would we now characterize a carrier? So I want you to move to the next. So this red colored protein here is actually a channel. So if something will diffuse through it, okay, it will not actually change its shape. Okay, you focus on the blue colored protein. Okay, you can see it's transporting the yellow colored substance from an area of higher concentration to an area of lower concentration. And as it transports the yellow colored substance, it actually changes its shape. Okay, so therefore the blue colored protein is a carrier. So again, facilitated passive transport makes use of either a channel or a carrier. Okay, the movement is from higher to lower concentration, and there's no need for the use of energy. If the protein does not change its shape as it transports the molecule, it's called a channel. But if it changes its conformation or shape, okay, as it transports the molecule, then it's called a carrier. Active transport, on the other hand, is the opposite of passive transport. This time, you're going to transport molecules from an area of lower concentration to an area of higher concentration. Gamay na lang gani kag love for yourself, ihatag pa jud di mo sa iya, ha? Okay? So, since this one is going against the concentration gradient or the normal behavior of molecules, this type of transport will now be requiring energy. That's why you can see that the ATP has to power, okay, the protein in the cell membrane for it to transport the molecule from area of lower concentration to area of higher concentration. And this protein now will be called a pump. Okay, a pump is used in active transport while channels and carriers are used in passive transport. So this is an example of an active transport. As you can see, the molecules are transported from area of lower concentration to higher concentration, and it's going against concentration gradient. So let's try to review what you have learned so far. Okay, the oxygen in the photo diffusing across the membrane without the help of a channel or carrier, then most likely that one is simple passive diffusion. Sodium diffusing from higher concentration to lower concentration through the help of a channel. So most likely this is what? Facilitated type of passive transport. While take a look at the active transport, as it transports sodium, okay, it transports it from an area of lower concentration, no, from area of, lower concentration to higher concentration. And there's the consumption of ATP, okay? So let's try to answer the following questions, okay? Requires energy, the answer is, no, you're supposed to answer passive transport or active transport. If your answer is passive transport, you have to be specific whether it is simple type or facilitated type. So again, requires energy. Oh, how daw, active daw. Oh, okay, fine. Next, oxygen and carbon dioxide diffusing across the membrane without the need for channel and carrier. Do not just answer passive transport. You have to be specific. Simple, passive transport. Very good. Okay, next. This type of transport is going against concentration gradient. The answer is what? Okay, active transport. Very good. Last, transport of molecule is from higher to lower concentration, and it requires a carrier or a channel. Okay, the answer is facilitated type of passive diffusion. Congratulations! So I would like to thank you again for spending time to listen to my lecture, and good luck with our first quiz. Okay, the quiz will be administered via Edmodo, and we will be sharing to your class the Edmodo classroom codes. Okay, and take note, there are two types of proteins that you can see in the cell membrane. So let's discuss them using this picture. The blue arrow, okay, is actually pointing to the integral protein. For you to easily remember the name integral, as you can see in the photo, the entire protein is integrated, okay, to the two layers of the membrane. So that's the reason why it's called integral. The other name for this protein is transmembrane protein because it traverses the two layers of the membrane. Okay, so you have first the integral protein or the transmembrane protein. Okay, the second type of protein, okay, is loosely attached to only one of the layers of the membrane. Okay, and since they are found at the sides, okay, or at the periphery, they will be referred to as the peripheral proteins. Okay, so I have a question and I want you to answer my question. Which among them is embedded within the cell membrane? The answer is integral. Which among them, okay, you have to analyze this one. Which among them can easily be extracted from the cell membrane? Because it is loosely attached to one of the layers of the membrane. The answer is peripheral. Very good. Okay. Which among them would require that the cell membrane has to be totally disrupted before you can harvest them? The answer is what? Integral. Okay, I can hear you answering. Even if I am just a recorded audio, I can hear you answering and you are guilty of it. Okay, so last question. Which among this one can act as transporter or channel? It's most likely the integral protein. So can you please give yourselves a round of applause, please, for getting the correct answers? Now, the presence of alveolar macrophages with brown colored pigments in their cytoplasm. So those brown pigments there are the hemosiderin. So if the pathologist will see the presence of these alveolar macrophages, the pathologist will have an idea that there was a previous bleeding or hemorrhage on this particular portion of the lung.